Alright, ladies and gentlemen, welcome back. So we're going to continue talking about microevolution, and today we're going to talk about a tool that has the potential to give us an understanding of whether or not evolution is actually occurring in a real population. And so this tool is this Hardy-Weinberg equilibrium principle. And what this principle really is saying that if we take the ideas of Mendelian genetics, right, and trace it through one generation to the next, the inheritance of those characteristics based on the way meiotic cell division occurs, how those genes get transferred from parent cell into gametes and then get recombined into offspring in the next generation. All of this is predictable. Okay, so if I look at that no matter what I do to the gene pool collectively, what I start with will always be the same ratio of alleles that I end up with in the following generation, assuming a certain set of criteria are met. So if I look at traditional Mendelian genetics, right? So we look at something like this. We start off with purebred red, purebred white. We combine them into generation 2. They happen to make all heterozygotes, which are pink. Well, when those pink flowers go ahead and reproduce, they have to produce 50% of their gametes that have that red allele, 50% of their gametes that have the white allele, so when they come back together to make the third generation, those heterozygote plus heterozygote produces this predictable 3-to-1 Mendelian ratio, right? 3 that have the dominant, 1 that has the recessive. So this particular ratio in this case is an incomplete dominant example, and so you get the 1:2:1 genotypic ratio. Homozygous dominant, heterozygote, and then homozygous recessive. Regardless, though, when I go to split out this entire gene pool's gametes, I'm going to end up with 50% red and 50% white. So when they recombined again in the following generation, the ratios remain the same. And that's what the Hardy-Weinberg principle is based on. The fact that the way that we create those gametes via meiosis and then recombine them during fertilization to make the next generation works the same way from the population level every single time. So what that means is that we can use Hardy-Weinberg to predict populations, to describe populations that are not evolving. And in doing so, the thing is that we're using this Hardy-Weinberg to predict populations that are not evolving, which also means that random mating has to occur, because if organisms select mates based on traits, that's sexual selection, which is a form of natural selection. It's also describing, as a result, populations where the allele frequencies aren't changing. All of this is a hypothetical population. If we look at real populations in the globe, in the ecosystems, Hardy-Weinberg rarely, if ever, actually happens. This equilibrium rarely ever occurs because the environment is going to be influencing who survives, who reproduces, and who doesn't. And so in most real populations, those allele frequencies and genotypic frequencies will change over time. There may be occasions where if I'm looking at a very specific gene locus, a very specific trait, maybe that trait isn't really changing over generations in that particular population, but generally speaking, most things are changing. So Hardy-Weinberg's idea is to create a hypothetical population, a baseline, if you will, for comparison to the real living populations that are out there. So how does this work? Well, there's a couple of equations to know. So really, Hardy-Weinberg is a mathematical model. So first off, when we use Hardy-Weinberg, it only works for organisms that are diploid. Okay, so that means that they've got 2 alleles for every trait. Think about your chromosome structure. Think about your genetic structure, right? You've got 2 sets of chromosomes, 1 set from your mom, 1 set from your dad. So each parent has given you an allele for every trait, a letter for every trait if we're going to use those conventional shorthands like you do with your Punnett squares, right? So every organism is diploid means that they have 2 letters or 2 genes from each parent, and it's the interplay between those genetics that then give you the physical structure that you have, or the phenotype, right? So the number of alleles at each locus is the total number of individuals times 2 at the population level. So if I have 10 individuals, then that means I have 20 alleles for that trait in the population. Okay. Then I need to start digging into how many of those alleles in the gene pool in the population are homozygous versus, or excuse me, how many of them are dominant versus how many are recessive. And that's where I need to know who's homozygous and who's heterozygous. So if I'm looking for the number of dominant alleles, then individuals who are homozygous dominant are contributing 2 capital letters, 2 dominant alleles. For individuals who are heterozygous, that means they have 1 copy of each allele, and so they're only contributing 1 copy to the population. The recessive would be very similar. If I'm looking at homozygous recessives, then they're contributing 2 alleles. If I'm looking at a heterozygous individual, they're contributing 1. If I take this collectively, there are always 2 alleles at every locus. That's by definition a diploid organism. And so Hardy-Weinberg takes the information for dominant versus recessive and gives them variables to make this look like a math equation, where you've got P and Q. Okay, P is your dominant allele. What's the frequency of the dominant allele in the population? Q is the frequency of the recessive. And so if I look at this from a mathematical standpoint, the portion of my population that's dominant, in this case represented by P, plus the portion of my population of my gene pool that is represented by the recessive allele, in this case Q, if I take those frequencies and add them up, they're always going to equal 1 or 100%. So if I have a frequency in the population of 0.2 for recessive and 0.8 for the dominant, 0.2 plus 0.8, add them together, that gets me the 1. Or think about them as percentages. I have 20% of the population that is contributing recessive alleles, 80% of the population is contributing dominant alleles, and so collectively together, that now constitutes 100% of the gene pool. So let's look at this with a real example. So here I have a wildflower population. I have 500 plants in the population. 320 individuals are red. So let's go back to that red, pink, white example that I showed you on a couple of slides before this. So if they're red, that means that I know that they're homozygous dominant based on that phenotype. So that means that the 320 individuals have to be contributing 2 copies of the allele. So that's where this is coming from right here. So 320 red, based on the way this inheritance works with incomplete dominance, all those red individuals have to be homozygous dominant, which means they're contributing 2 copies of that dominant allele. As a result, from those individuals collectively, 640 recessive alleles are being contributed to the population. I also have 160 pink flowers. And again, because this is incomplete dominance, that means I know that those pink individuals are heterozygous. And so for 160 pink, they have a single copy of the dominant allele. So they are contributing 160 dominant alleles to the population. If I look at those dominants, that means I've got 640 for my homozygous dominant, 160 for my heterozygous. As a result, 800 alleles in that population are dominant. Go back to then talking about recessive. Well, I have that 160 pink, aka heterozygous, and so they're contributing 160 recessive alleles to the population as well. And then I also find that I have 20 white individuals. Again, because of this being incomplete dominance, that means I know that they are homozygous recessive, so they have 2 copies of the white allele and thus are contributing 40 alleles of the recessive type to the population. Add that together, the 160 from the heterozygotes plus the 40 from the homozygous recessives, and I have 200 recessive alleles in the population. Now, when we talk Hardy-Weinberg, we're talking about this as a frequency within the population itself. So go back to those 500 plants in the population. They're diploid. So that means that they have 2 alleles for every trait. So 500 times 2 is where this 1,000 in alleles is coming from. And then I want to know what's the frequency of that allele in my population based on the total number of alleles present. So if I take that 800 dominant alleles that we calculated back here, divide that by the total 1,000 alleles present in the population, I get 0.8 or 80%. The recessives, 200 in the population out of 1,000 gets me the 0.2 or 20%. Now what I will say is that they will often leave Hardy-Weinberg as decimals, not percentages, so just be careful when you're doing practice problems or if this were to show up on a test question, what they're looking for. Are they looking strictly for the frequency, in which case you'll probably leave it as a decimal, or are they asking for a percentage, in which case you can convert it that way. Regardless, if I go back to Hardy-Weinberg, P plus Q always has to equal 1, or the percentage of P plus the percentage of Q has to equal 100%. So 0.8 plus 0.2 equals the 1, or 80% plus 20% gives you the 100%. You can't have more than 100% in the population. You shouldn't have less, otherwise you've missed individuals. So if our wildflower population pool remains constant from one generation to the next, meaning we always stay at that 80-20 ratio, then I know that the only thing contributing to the gene pool is Mendelian genetics, right? Mendelian processes alone can't alter the frequency of alleles, can't alter the frequency of genotypes. And so long as that 80-20 ratio is happening, then the only thing that's going on is simple meiotic division, creating gametes, and then fertilization to make the next generation. However, while we're on this topic, if anything were to cause those ratios to change, then we can start digging into what's going on at an environmental level that might be causing evolution to be occurring. Other piece to this, all right, so the example I just gave you with the red, white, and pink flowers is really a nice one because it's very clear which phenotype is connected to which genotype. But if you remember your Mendelian genetics, most traits don't work like that. They're simple dominant versus recessive. So either they're showing the dominant trait that you can physically see, in which case they could be homozygous dominant, or they could be heterozygous, or they are homozygous recessive, in which case you would see a distinct phenotype there. So if we want to start looking at the population from that standpoint, then I have to start taking into consideration who's homozygous dominant, who's heterozygous, who's homozygous recessive, and then I can start teasing out dominant allele frequencies and recessive allele frequencies. So the P plus Q equals 1 is one formula that comes out of Hardy-Weinberg equilibrium. The second is this one, and all it's stating is that P squared represents the homozygous dominant individuals, right, because each of them is contributing twice to the population. Heterozygote individuals are P times Q times 2 alleles that they're contributing collectively. They're contributing one that's the P, one that's the Q, and then Q squared is homozygous recessive. Okay. So again, if we look at these frequencies and pull these out, so long as that 80-20 relationship stays consistent, then evolution isn't happening, at least in this population, for this trait. okay? But if these ratios change, then that can be an indication that something is being selected for or against. So if I take that same idea of that 80-20, right? P is 0.8, Q is 0.2, and I pull it into wanting to know the frequencies of the actual genotypes, then P squared is 0.8 squared, or 64%. Heterozygous is 2PQ, or 32%, right? 2 times 0.8 times 0.2. Q squared, or 0.2 squared, gets you 4% of the next generation being that homozygous recessive. So keeping in mind that Hardy-Weinberg equilibrium is just a tool to understand how populations would look should they not evolve, and also keeping in the back of your mind that most populations don't follow this, there are 5 conditions that a population would need to meet in order for it to actually fall under and work as Hardy-Weinberg predicted. AKA, what does it look like for a population if it's not evolving? And there are certain conditions that have to be in place for this to be true, for it to not actually end up evolving. So first and foremost, in order for Hardy-Weinberg to work, the population to not evolve means this population has to have an extremely large population size. The smaller and smaller that population size becomes, the more likely that changes in the fluctuations of allele frequencies have nothing to do with evolution and more just to do with chance. And that's where we'll talk a little bit later about genetic drift. So a Hardy-Weinberg population must be large enough so that the genetic drift doesn't occur. We also can't have any gene flow. So when we talk about gene flow, we're talking about the transfer of alleles between populations, aka I can't have any individuals moving into my population during that period of time because they're bringing alleles with them, and bringing those alleles will adjust the ratio of alleles in the next generation. And the same thing would go in the opposite direction. They can't be leaving either. And so gene flow really is just the immigration and emigration, the moving in and moving out of individuals. I can't have that happen because when that occurs, that is naturally going to change the frequencies of alleles in those populations because I'm changing the number of individuals and the alleles that they're bringing with them or taking away. Also, can't have any mutations. Okay, similar to that gene flow adjusting ratios, mutations would do the same thing. Okay, so I can't introduce or remove genes by mutation, otherwise that will influence the ratio of alleles. It also has to be completely random mating. If an organism is choosing a mate based on some sort of preference, that's sexual selection, and sexual selection is a form of natural selection. You're choosing who to mate with, so you're choosing whose alleles are going to move on to the next generation based on preferences from a mating standpoint. And then overall, I can't have any natural selection, period. Whether it's based on sexual characteristics, whether it's based on environmental conditions, every individual in that population, for it not to evolve, for it to fit Hardy-Weinberg, has to be able to have the same success or failure at survival and reproduction as every other individual in the population. And that right there is where most populations end up failing Hardy-Weinberg in true living nature because there's going to be differential success in most populations. And so for Hardy-Weinberg to exist and not change those allele frequencies from one generation to the next, then everybody has to have an equal chance of surviving and reproducing and passing on those traits.